Today, the topic is about very specific drug screening dataset. So we're aiming for FAIRification of drug sensitivity screening data to improve the translational medicine. So my name is Jing Tang. I'm an associate professor in medical bioinformatics from the University of Helsinki. And this work has been jointly done together with my colleagues from Assays, as well as funded by the project called EOS5. So the agenda for the webinar today is 4 parts. So first, I will give some introduction to translational medicine, and then I will talk specifically about the motivation of why we should work on the FAIRification of drug sensitivity assays. After that, I will give a very short demo of MICHA, which is the web application to facilitate the FAIRification. And then I hope to have some discussions with the audience. So if we go to the current drug discovery process, basically it involves 3 components, starting from target identification, and then with the lead optimization to choose some prioritized compounds, and later for patient stratification in a typical clinical trial. And if you look at the whole process, may take on average 10 years for a compound to reach to the market. So basically to reach to the patient for the treatment of their diseases. And if you look at the cost efficiency in drug discovery, which is shows that the cost of typical drug discovery has been increasing all the time. And if you instead look at the successful rate of those compounds, which have been finally approved by the FDA, for example, it has been, pretty low in general. Of course, it depends on what type of diseases a compound can be used for the treatment. But on average, if you look at all the indications, the probability of success is below than 10%. So that basically shows the difficulty to facilitate the drug discovery based on the knowledge from translational medicine. I believe most of us have been already well informed about the challenge of doing translational medicine, and then maybe kind of critical to ask a question: why has it been so difficult? So basically, there are a lot of explanations for the for this scenario, for this kind of scenario. So of course, we're not going to cover all the potential factors that may affect the translational potential of the knowledge to drug discovery. And I would like to mainly tell some stories from a pharmacological point of view to give you some examples why it has been so difficult to identify an effective compound for treating a disease. So this is the paper that was recently published around 3 years ago. So basically, it is a cover story of a journal of Science Translational Medicine. So basically, it shows the challenges for understanding the mechanism of action of the compound, even though many compounds are already clinically approved or have been tested in the clinical trials. So this is an example which was shown in that paper. So it's a PF3785309 drug. And if you go to Google, for example, or go to any chemical compound supplier website. So it reads that it is a very potent ATP competitive inhibitor of a drug target called PAK4. And actually, this is a quote that was copied paste from SELEC website, which is a major chemical compound supplier. So it says that it is a very potent PAK4 inhibitor with IC50 of 1.3 nanomolar. So, and it has been shown to be very effective to induce cell growth inhibition in the HCT116 tumor model, which is a cell line. So if you go to the clinical trial website, for example, the clinicaltrials.gov, and actually this compound has been entered into phase 1 clinical trial. But what is surprising is that actually this drug is not an PAK4 inhibitor. So why was that? Because the author of a paper, they basically show that if you knock down PAK4 in the cell lines, and if this drug is a PAK4 inhibitor, if the target is removed, then basically you're expecting a drug is not going to work, right? But actually, that was not the case, as it was shown in these two figures. So no matter whether you knock out this PAK4 or not, actually the drug is still very effective. They have tested the same hypothesis in both of the cell lines, so A735 and also this HCT116 cell line, which was exactly the same cell line that was mentioned here. So they found both cases where the drug is still kind of super effective, irrespective of whether the PAK4 is present or not. So basically, this experiment suggested that actually there must be some other targets which are unknown that are still involved in the efficacy of the drugs. So basically, that means even a drug has been tested in the clinical trials or even in the clinics. So we still have a very little understanding about what are the other potential targets for treating cancer. So this is another complexity of a typical drug, especially for small molecules, which are often targeted the drug. So we probably don't necessarily assume that the drug is very specific, because for most of the cases, if you look at targeted drug, for example, like kinase inhibitors, their targets are actually very kind of promiscuous, meaning that there are many targets for the same compound. So this is the figure shows example of the two compounds, axitinib and sunitinib. So actually, this is also experimental data. So based on binding affinity assays. So that shows that actually these two compounds are multi-targeted, and also they are having a lot of targets which are overlapping. So imagine if you want to know exactly what is the function of those targets, it's super challenging to deconvolute those interactions in their drug target profiles. So polypharmacology is actually the reality, it's the norm, not an exception. And so let's show that it's super important to understand the complete profiles of drug-target interactions. Unfortunately, for this kind of knowledge, we still have a very little understanding due to the limitations of binding affinity assay, for example, that it's not easy to profile all the protein targets in a proteome for a particular compound. So what I have just described are mainly from the pharmacological point of view, try to explain why it is so difficult to develop a successful compound. So the reason because that we have very little understanding about the drug target profiles, and also that may affect your clinical trial result. And on the other hand, if we look at the patient, we also very kind of puzzled about a common pattern that not all the patients respond to the same treatment. And if you look at the cancer therapy, so there are 5 pillars of cancer treatment. People here are more enthusiastic about the targeted therapy and immunotherapy. But actually, if you look at the efficacy of the most recent immunotherapy, it is quite disappointing responding rate, suggesting that only 20% of patients respond to immunotherapy. And on the other hand, even though a drug is very effective when it was first given to a patient, but the patient can easily develop this type of resistance, as it indicated by this particular example, which is a patient of melanoma treated with vemurafenib. So as you can see, even though the initial response is surprisingly good, but the patient develops the resistance, so that this drug is not working anymore. And if you look at the many clinical trial results, you can see this type of converging effect towards the end of the clinical trials, that you don't see a strong benefit anymore when the time goes on. So this is try to give you a summary about those grand challenges in translational medicine. So basically, we're trying to put those pieces together, and mainly asking 4 critical questions. For example, we want to know which patients are potentially non-responders. We don't want to give non-responders a cancer drug. We don't want to just give it for nothing, even we know already the patient is not going to respond. And then we also need to understand why patients so easily develop the resistance. And from pharmacological point of view, we also want to know better about the target profiles, so that we can deconvolute the efficacy as well as the side effect of a particular compound. And then with the knowledge that we have been accumulating in the process, we hope we can suggest or try to predict the more effective treatment to overcome the drug resistance. So in the end, we would like to deliver our promises for drug response prediction. Ideally, maybe in 10 or 15 years from now, we hope to be able to predict the best treatment option given a patient's clinical and molecular profile. And basically, if you transform it into a machine learning or artificial intelligence term, That means we try to make a predictive model, trying to take the genotype of the patient as the input, and then trying to predict the pharmacotype, which means that suggestion about the best treatment for this particular patient. And the model ideally should be interpretable as well as testable, especially for cancer. If we have quite many cancer cell lines or mouse models, we always want to test the prediction first in those preclinical models before we go to clinical trials. So that's the reason why we need to generate the drug sensitivity assays, which is the major topic of today. So I have spent quite a few minutes trying to give you the rationale why drug sensitivity data is important. So because if you want to develop any predictive models, we need the training data, right? So without drug sensitivity assays, it's super difficult to infer what might happen if a drug is given to a patient. So that's why people nowadays, they are trying different type of cancer cell models, which are derived from actual patients. So basically, as is shown here, we are interested in patient-derived materials, such as cancer organoids, and we need to culture them so that they can be basically effective, considered as a mimic of the actual patient. And then we're trying to do the sequencing as well as the drug testing for this type of cancer samples. And then the data will be integrated into a predictive model so that we can know better about the drug response biomarkers, which will be utilized as a rationale for the clinical trial design. And this is an example of this type of personalized medicine drug discovery platform, which has been up and running at the University of Helsinki and the hospital. So for the moment, there are multiple cancer types which are available for this type of testing. And I here give you a few examples, including leukemia, pancreatic cancer, and ovarian cancer. So the samples are taken from patients and cultured in the lab, and then we can run the drug testing and the genome and molecular profiling. So that data has been used for producing quite promising results for the clinicians about the potential new drugs for treating the cancers. And here I just give you an example of how this kind of platform has been useful for drug discovery. So this was a study that was published a few years ago in Nature. So basically, the key finding is that they found a new indication of existing drug called axitinib. So axitinib has been approved as a VEGFR inhibitor for renal cancer. So this is the structure of its drug, and this is how the drug has been interacting with the VEGFR protein. And using the drug testing platform, the drug has been used for testing the leukemia patient samples. So from here, we can find out that this drug is actually quite effective, affecting the cancer cells from leukemia patients. So ponatinib is another drug which is also effective, and this ponatinib is already approved as an ABL inhibitor for leukemia. So you can see this axitinib is also is much more effective than the other compound. Also, as shown in this figure, that the axitinib has a strong dose response inhibition, although it's not as much as ponatinib, but it also shows very good promise. And later on, the researchers have been using the crystallization experiment to confirm that indeed this drug has been bonded to the ABL-T315 drug target. So ABL-T315 is a gatekeeper mutation, which is a cancer target for leukemia. So then the drug has been tested in more samples and later on even to the actual patient, which shows very strong efficacy. So basically, this story shows the value of using drug sensitivity assays for discovering the compounds which have not been known before as a drug repositioning application. So taken together, we believe that the drug sensitivity screening is very important. It may facilitate the drug discovery, as shown in the previous example of this axitinib story. So the whole process, if it is really in a very good quality control, then we're hoping it will bring a great promise for personalized medicine discovery. But actually, I think the reality is that we're not quite there yet due to many potential issues of the drug sensitivity assay screening. That is also a kind of a bottleneck or like a challenge to really apply the result of the drug sensitivity assays for drug discovery. I will give you a few examples about what might be the issue. So the major issue is about the reproducibility, which has been always a concern for a typical biological experiment. So there's no exception for a drug sensitivity assay. So this figure, which was reported in Harper around 10 years ago, they just compare the drug sensitivity data from two major drug screening studies. One is called CCLE, so CCLE is here, and the other is called the CGP. So they are comparing the correlation between the drug sensitivity data from the two major studies on the same compound. And actually, the correlation is not on the top, particularly for a subset of a compound. And you don't see a correlation at all, which shows the difficulty to utilize any of the results for your next experiment or biological investigation. And a few years later, and in a more recent paper, so they look into more details about what are the effects of those experimental parameters made bring to the reproducibility. So, for example, they compare if they are using different type of assays to measure the cell viability, how different the result can be. For example, if you use the direct cell counting versus the cell titer glow assay, which measures mainly the metabolic activity of the cells. So, and then you can compare the dose response curves of the same compound at the same concentration. You can see some of the compounds have very good consistency, meaning that the measurement techniques doesn't affect the dose response, but some of the compounds actually has more divergent curves. And they also evaluated some other parameters, for example, like different dose ranges, and also different data processing algorithms, counting. And they found that some of the experimental parameters are more dominant in affecting the reproducibility than the others. So these papers, they are reporting those results that are indicative of reproducibility issue is our motivation. So we think if we are able to make this kind of annotation of experimental protocols more transparent, then we're aiming for more reproducibility data analysis. So the idea is that we are trying to make the drug sensitivity assays more fair. So fair is a very specific term, trying to improve the openness of the research data, which means that we need to make the data findable, accessible, interoperable, and reusable. So that's our belief that if we can manage to make this process a bit more systematic and easy to implement, then people may adopt this type of FAIRification for their own drug sensitivity assays. So that's why we're developing this MICHA. So MICHA is a short name for the minimal information about chemosensitivity assays. And the aim of the MICHA is to verify the drug sensitivity assay protocols that are critical for evaluating the data reproducibility. In MICHA, there are 4 major components, which are compounds, cells, assays, and data processing. Why we want to focus on these 4 major components? Because in a typical drug sensitivity assay, we need to understand about the target, about the compound, and also the materials, and also also in the end how the data has been pre-processed. So for example, for the first component, which is compound, we want to know what are the targets of the compound. And for the samples, we want to know the characteristic of the samples, and assay protocol, as well as the data pre-processing and quality control method. So we're trying to develop MICHA as a web server. So it's not a simple like a checkbox that you have to fill in, but it's more like a web server for you to make this verification as efficient as possible. We call it as a semi-automation. I will show in a few minutes about what do we mean by this semi-automation. So this is the so-called the flowchart of MICHA. And if you go to the website, it also shows some summary of the data that is available. So now I'm going to give a demonstration about MICHA. So basically, we need to go to this website and to check how the MICHA can be utilized for automation of the drug sensitivity assay verification. So I'm going to click on this. Okay. Now I hope you are able to see the website. So this is the MICHA website. So when you go to the website, you will see this welcome information, and this shows the ID of MICHA. So if you close that, basically it's a search window. But for now, we don't go to the search window. We just want to show how to utilize MICHA for verification of your data. So normally you have to upload input file. So here I give you also a template of the input to MICHA. So you can either download the templates or upload the templates directly. So for now, I'm going to first to show what is the template, what information is included in the template. So actually, okay, I need to download templates. It's already here. So I guess you don't see the Excel table. I have to reshare again. So after you download the template, open it, and this is actually the content of the template table. Basically, it has two columns. The first column, you have to put the name of the compound. The second term is called entry key, meaning that you have to also put the entry key ID for those compounds. So basically, this table just show how your compounds look like. So you just need to give the name of the compound as well as the entry key of the compound. So imagine you have tested this many compounds, and then you want to verify your chemosensitivity assays by first uploading this input to MICHA. So now I will go back to MICHA. So actually, you don't need to re-upload again. So, but anyway, you don't need to. You just need to click the button here called Upload Template to upload the file that you have just seen. So basically, after you upload the input, then what MICHA is trying to do is to go to all the major chemical biology databases, trying to fetch the annotation of those compounds that you have uploaded. So for example, what you have seen here to show is the primary target of the compounds that you have uploaded. Also, the other potent targets, which is more like a secondary target of the compound, and other information such as the clinical trial stage and cross references of the compound. For example, you can go to Findable or Campbell to get more information of those compounds that you have selected. So as you can see from here, MICHA is more like a data integration server, trying to fetch all the available information for a compound from multiple major databases such as Campbell or PubChem. So this is the first step of MICHA, which is the annotation of your compound. And then after you're fine with it, then you can go to the next stage. So as I show in my slide, there are 4 components in MICHA. So compound, samples, reagents, and data. So the second component is the samples. If you click the tab here, that means you need to annotate your samples. So what do we mean by samples? Usually it's about your cell line names. For example, here I just give example of 2 cell lines, and I can put more information about their passage number, modification, and if the samples are patient-derived, you may also input the clinical phenotypes of those samples, such as the age, diagnosis, sample material, and etc. You can also create a new entry depending on how many samples you have. So imagine you have more cell lines, and you can put the name of the cell line and put all the information, the ID, and source ID as well, or some other information of the sample. You can click Add, then you have more in this table. So that means this table can be used for a source of information about those samples. And then if you are satisfied with your sample annotation, then you can go to reagents. Reagents basically means that what are the experimental protocols So here, I just show example of a typical cell viability screening that you have to specify what is the cell viability detection technology, what kind of medium it is, and the plate type, number of cells at plating, time of treatment, whether it's 24 hours treatment or 40 hours treatment, 48 or 72. So you can create all the information through the table here by creating a new entry or updating the existing one. So this is the third component of MICHA. And the last component is called data, which is mainly about data preprocessing. So basically, you can put information about what is the metric of your dose response, whether it is IC50 or something else. For example, it can be AUC as well. That is based on the area under the curve of the dose response. You can also put other information, such as the minimum concentration or the maximum concentration of your dose response curve. That is also important to be annotated. So in the end, if you are fine with all the components, you are filling all the necessary information, then you can click the download. So after you click the download, there will be a new table, new Excel table. You can save it. So basically, after all the information that has been recorded, then you can see, okay, I will stop sharing again. And so now is the table after you have filled in all the information. So as it shows in the website, there are also like 4 tabs corresponding to the 4 components in MICHA, which is compound, samples, reagent, and data processing. So in the compound tab, it saves all information about the drug targets and other important details of the drug. And in the sample spreadsheet, you can see what I have just inputted as the name of the cell lines and some other information. So the information are all saved in a single Excel table, which contains the 4 components. And this table, I think it will be very helpful for you to identify those critical experimental protocols. For example, like what is the name of the detection technology? And also importantly, if you have the table, you can basically easily compare different studies because everything will be standardized and harmonized this type of tabular format. So this just shows some example of how MICHA is going to help you with the annotation of experiment. Okay, I hope you can still see the slide, which is at the page of demonstration. And then I will give some more like overview of the capacity of MICHA and also like use case of MICHA. So first of all, I think MICHA can be considered as a drug target annotation tool, as I have just shown in the demo, that if you have a list of compounds and you're interested what the targets of the compound, what are the physiochemical properties of the compound, then you can go to MICHA to fetch all the relevant information in the server. So what MICHA has done already is that it has been already pre-compiled list of compounds, and there are also targets. And if you go to the comparison of the coverage of the drug target interaction information, actually MICHA has a top coverage of the number of targets that you can find as compared to the other major databases, which is not surprising because the idea of MICHA is to integrate the information from multiple databases. So that's why MICHA can be considered as an integration of the drug target information. On the other hand, MICHA can also facilitate the catalog of the drug sensitivity studies. Imagine that multiple studies have used the MICHA for annotation of their compound. Then we can easily compare the differences of the protocols between multiple studies. We have done this type of comparison already for those major chemical screening studies, for example, like GDSC, CCLE, and CTRP. So we can easily compare the differences of the experimental protocols, and then we can have a better idea about what the techniques that are more commonly used, and how these techniques are giving the different results in the drug sensitivity data. In addition to cancer, which is of course a major disease that has been studied in drug sensitivity assays, we have also curated the more recent COVID-19 drug sensitivity assays, including those that are shown here, which are also useful for comparing the differences of their experimental details. And what MICHA, if we have all the protocols that have been curated and FAIRified, then we can easily compare the overlap of the compound and the cell lines. For example, this shows the major drug sensitivity assays in cancer. We can have a clear overview of the overlapping compounds and the cell lines. We can also have the overview of the experimental protocols comparison across the different major cancer studies. So the summary is that we propose a novel workflow called MICHA, which has a website and up and running for the FAIRification of drug sensitivity screening protocols. And MICHA can provide an integrated platform to obtain drug screening assay annotations, drug target profiles, and other pharmacological information in an easy and a fast manner. And through the use of MICHA, we are able to FAIRify drug screening protocols related to cancer and COVID. Then these FAIRified protocols can be made comparable and informative for designing a new experiment. For example, you can check what are the major techniques used, then you can try to follow the same type of protocol for your own experiment. So in addition to MICHA, I have also have a research group in the University of Helsinki, where many working on the data analysis of translational medicine. We are developing more resources on drug sensitivity data integration. For example, we have built a database called DrugCom, where you can find more information about drug combinations. And we have developed also software to analyze drug combination data, as well as the drug dose response curve data. So MICHA is one of the informatics tools that are under development. Hopefully, these tools can be used together for facilitating the translational medicine drug discovery. And this is actually a joint work together with multiple institutions within ERTRIS, for example, like the NIH, NCAS, and also FIM from the University of Helsinki, and other institutions, also partners within the EOS Live Consortium. So with that, I'm very happy to take any questions and comments about MICHA. Thank you.
So before any question comes in, can I ask Jing, are there some R&D companies or drug screening companies using MICHA platform for their development?
So far, yeah, the major users are from those institutions that I have mentioned in the final slide. Those are like participants of the MICHA, which have also utilized the MICHA for their own drug sensitivity screening data. We're hoping with the webinar we can attract more users to use MICHA to provide some comments and feedback about how to use MICHA more easily for annotation of their data sets. We're hoping more people and more institutions can use this MICHA for annotating their data set. because anyway, it will be important to make the drug sensitivity assays like transparent and open, so people will know how to compare the different protocols.
One question just came in. So what is the novelty of MICHA compared to other softwares?
And the novelty of MICHA is that it's the first implementation to verify the drug sensitivity assays. It's a very specific tool. It's used for only annotating the drug sensitivity assays, and the software is it can be used as multiple purposes. For example, if you want to identify the targets of the drugs, you can use MICHA. You don't need to go to those individual databases to fetch the information about the compound. Instead, you can just go to MICHA and input the name of the compound and the entity key of the compound, and the MICHA will do the data integration automatically for you. So it's more like a web server, and which can give you information quickly and efficiently.
Yes, we have another question just came in. How do you upload your own data into MICHA? I think you showed one upload, but
yeah,
can you upload your own data?
You can just prepare your data in the same format as I just showed earlier. Basically, you just need two columns. First column is the name of the compound. Second column is the entry key of the compound. So after you prepare these two columns, you can upload it into MICHA, and MICHA will find the targets and other information of the compound for you. That's the first step of the FAIRification, which is about compound. Then you can do the same thing for the other components, such as cancer samples or experimental protocols.
I just wanted to ask, you mentioned the entry key of all the compounds. So is it associated with all the compounds that are being generated, or is something which you create for a compound?
Actually, entry key information is quite standardized. You can go to any major chemical compound databases, such as Campbell or PubChem. You search a name of compound, and the database will give you the entry key. So basically, it's based on the structure of the compound. It's more like a hashtag or like encryption of the chemical structure.
So another question is, any future improvements on MICHA, or is there any development work going on for this platform?
Yeah, our vision is that MICHA can be more like a community-driven project. So far, there are only like 6 or 7 participants developing MICHA, but we're hoping more people can join. We want to develop this web server more user-friendly, and also as a community effort, so we can try to annotate as many as possible different studies. We need this kind of curation type of work to be done in the future.
So one of our attendees has tried the MICHA platform, I think right now. They're saying that it's not giving the icon to upload it.
Icon.
Yeah, the icon which you can click to upload the data. Is it possible to show it in your browser, maybe?
Okay, let me see. So now you are able to see the web server now?
Yes.
So you can upload the file, right? You click on choose file. For example, I just download the template, then I'll upload again. So this should be the, and you click submit. So it will give you basically the information about the compound, and then you can start working on the other components. As I think, I mean, for the other component, it's more manual work. You have to input a lot of information because it's less automatic compared to the compound. For compound, we have a very good source of information, but for others, you can see a summary of those information that is available in MICHA, like different type of assays, and so yeah, then you can go continue from here.
Okay, thanks. Yeah, so I think it works for Kasim.
Yeah, If you have any questions, actually we have this glossary tab. So if you are unsure about what needs to be input there, you can find the meaning of those terms, different type of properties of compound, different type of experiment. And for some of the terms, we also have the BAO. BAO is like a bioassay annotation, which can be also used for cross-link to the other databases. So it's a quite comprehensive information about the terms. And here in the protocols, it shows those FAIRified protocols by MICHA, for example, like from different institutions, what the compounds are they using, what are the experimental protocols they have adopted. You can easily compare the differences and the similarity across different studies. And we also provide the API. So I missed that. But anyway, you can go to the API and fetch information from database more directly.
Oh, that's good.
Yeah.
There's one more question. Does it accept canonical smiles?
Yeah, we're thinking about that, but we feel like NGK ID is more standardized and has less ambiguity. So that usually is more preferred ID for the compound. So that's why for the moment we only allow NGK, just for the for the ease of the data integration, because with the NGK you can easily go to different databases to fetch exactly the same compound, not something else. So we want to make sure the high quality of the annotation.
No, I think our attendees are saying thanks to you, and they're saying nice tool. So
yeah.
that's good. So thank you, Jing, for today's webinar, and thank you all for attending.
Yeah, thank you.